Good morning, everyone, and welcome to Emerald Ash Borer University. This is Robin Osborne from Michigan State University, and my EAB University colleagues, Professors Cliff Sadoff and Elizabeth Barnes from Purdue University, and Amy Stone from Ohio State University Extension, are happy to share with you today's webinar entitled What We Know So Far About How Feeding and Mating Are Related to Spotted Lanternfly Dispersal Behavior will be presented. Our presenter today is Dr. Tom Baker, entomologist in the Department of Entomology, Center for Chemical Ecology at Pennsylvania State University. Dr. Baker has been performing research in the field of insect chemical ecology for 48 years. He was elected Fellow of the Entomological Society of America in 2009, was named University Distinguished Professor at Penn State in 2010, Fellow of the American Association for the Advancement of Science, and in 2012 received the Silver Medal Award of the International Society of Chemical Ecology for career achievement in his field. Over his career, Dr. Baker's research has helped uncover the mechanism used by flying insects to locate odor sources from a distance, as well as the olfactory pathways insects use to discriminate among odors. He has always looked for ways to apply knowledge about insect pheromones and other attractants for use in mating disruption and for detection and monitoring of agricultural insect pests and invasive species. Over the years, this work has gained a wide scientific readership, with 211 original scientific research papers published and 32 review articles and book chapters providing his overviews and summaries of his field. We welcome your comments and questions today, so please feel free to type them in the chat pod or the Q&A pod, and Dr. Baker will be answering them right after his presentation. Tomorrow you will be emailed a link to a short, voluntary, confidential survey that we hope you will take the time to fill out. It helps us bring these free educational webinars to you. The email will also include our presenters' contact information, as well as information on how to obtain CEUs for viewing this live webinar. This webinar is being recorded and will be available for viewing later today on the EAB University page of the www.emeraldashborer.info website. Thank you, everyone, for attending today. And Dr. Baker, please feel free to start your presentation.
Okay, thanks, Robin. Are you recording? I'm not sure I can see from my screen.
Yes, I did hit it is on record.
Okay, great. Well, thanks for inviting me to talk to Emerald Ash Borer University. I did a lot of research on Emerald Ash Borer, and I'm a Michigan State PhD and spent a lot of time in East Lansing and all over. And we were up there in Brighton, the early days of the quarantine before it blew up, the population blew up. And so I'm quite familiar with emerald ash borer. And now I would like to tell you something about our research on the spotted lanternfly, and specifically on what we've learned about the flight dispersal behavior of this pernicious, weird insect. So these are some of the people that were involved in this research. Julie Urban, who's heading up a massive USDA FIFRA critical applied research grant, and her crew was involved in this. But my crew, back starting from a long time ago, 2015, we've been working on this insect. This is a plant hopper, even though it's called a fulgurid. I never thought in my life I'd ever be working on a fulgurid bug, these peanut bugs that used to fascinate me. You see them in the tropics. They're normally tropical, but this weird guy is in the temperate region and does quite well even in sub-freezing temperatures. seem to come from China or one of the Asian countries there. It's invaded Korea and Japan before it ever got to the United States. And so it's a plant-sucking insect, and it arrived in eastern Pennsylvania. This was the map of the spread of spotted lanternfly in eastern Pennsylvania from 2014, when it was discovered, through 2017. So you see this area, and it's down here in eastern, southeastern Pennsylvania. Here's Philadelphia. And we do a lot of mushroom work, mushroom fly pest work down here also. Lots of mushroom growing down in this area. But this is where it started. And in 2019, this is the map where the quarantine zones have spread to in southeastern Pennsylvania, into Maryland, Virginia, and Delaware. This was 2019. And these were yellow counties are fines, but not an infestation necessarily, 2019. So in March of 2020, you see, okay, those counties, and then it's over by Pittsburgh now. Okay, and then these little dots that maybe don't show up so well on your screen, those are counties where there were fines, but they say there's no infestation. So that was March of this year. And now you see in September, here we've got an infestation in Connecticut and in parts of Staten Island, New York, and more dots where there were fines. Okay, and I understand Ohio has a lot of worries about this right now. I'm a native of Cleveland, so back in the day, so I know Ohio pretty well. So this is yellow dot is where we were working. And this is, again, in this region of Pennsylvania, right in here. So that was our initial field site area in 2015 when we started working on this. And this was the initial discovery site, which our field sites were only 3 kilometers, 6 kilometers, 4 kilometers, 6 kilometers in different directions away from this initial discovery site. These guys, the first instars, look like this. They're tiny little guys, and they are maddeningly hoppers. They hop all over the place, really weird. And the fourth instar gets on this episomatic coloring. They are toxic. Olanthone is one of the distasteful compounds and toxic inside these, and it comes from the Ailanthus tree. And the preferred host is a tree of heaven, Ailanthus altissimum. And these are some of the photos I took in 2017 on Ailanthus. Here you see fourth instar and some adults at the same time. Here's a fourth instar and an adult. Okay, on Ailanthus. They can look like this. Even in 2015, we found accumulations at the base of Ailanthus trees that look like this, and they squirt out this honeydew that creates black mold and all sorts of weird bacterial growths at the base of these Ailanthus trees. They love grapes. If you're a grape grower, I know, and Robin said their grape growers are really scared about this pest. California, we have contacts in California, and they're really worried about the dispersal of these guys. They also will feed on apple, and they don't feed on it so much, but here you see a lot of them in apple in eastern Pennsylvania. Pennsylvania's forest wood products industry is really concerned. This is black walnut, and these spotted lanternflies, once the population is built up over the years here in Pennsylvania, you would see them much more frequently on black walnut, and they feed very nicely on black walnut trees. And Pennsylvania's wood products industry for hardwoods is a really big deal. 70% of Pennsylvania's acreage over the state is forest. So all of our research, I could summarize. I can't. I blocked out some of it. So I'm a sex pheromone guy. That's most of what I've worked on in my career. Let me minimize this so I can see it. I found no evidence that these lanternflies are using a pheromone or any other kind of attractant. I saw no behavioral evidence that there's any long or short distance communication of any type with either sound. There might be vibrational cues that they use, but I saw no directed movement of one individual towards another from any distance, even a few tens of centimeters, over all the times I was observing them and my postdocs and graduate students. Doesn't seem there's any visual response to each other, the individuals. So then there's no behavioral evidence that I saw that there was preferential orientation to Ailanthus altissima or to any other species of plant. And this is what we found in 2015 and 16. And we saw no flights in 2015 and 16. You just sit there and watch them and they're just walking around on the tree. And we didn't see flight dispersal behavior of any significance. One small high-flying swarm of about 25 individuals we saw on one day in September 2016. And that was it. So very little behavioral activity of any type that we could see. But then all hell broke loose with incredible mass flights in September of 2017. And we wrote about this in our paper in 2000, published in 2019, about the flight dispersal, mass flight dispersal behavior in 2017 and the seasonal activities of the adults leading up to that. So this is what this flight dispersal behavior looked like on the first days when the alarm bells really went off in eastern Pennsylvania. So this is like an amazing locust swarm. You see them taking off from apple trees and flying generally upwind. Wind is from the left here. Okay, massive numbers. And there were newspaper articles all over the place. And this is from the Reading Eagle. The Reading was near the epicenter of this. And the people were reporting thousands, tens of thousands of lanternflies landing in the Walmart parking lot in Bechtelsville. And the adults were so numerous, they were walking by the thousands, trying to walk into the front door of the Walmart, and people were just crushing them and slipping on them. There were so many of them. And this one woman was quoted, "Honestly, we could go out in flip-flops and kill them." And there were gas stations and that were just diluted with these adults landing in there. And people were saying, well, who knows where they're going? That's what Marianne Lieberman was quoted as saying. So here's a vineyard that we worked in near Reading. On this, I will not disclose the name of the owner. I'm not supposed to. At any rate, it looked pretty good in 2017. And you notice there's one telephone pole here. By 2019, it was completely obliterated. Okay. And there were still adult spotted lanternflies on the top of the grape post. But the vines were all killed, nearly all of them. So it's tremendous damage. And the adults are the ones that really end up sucking most of the sap and nutrients out of the plants. They just have so much honeydew they produce, and it just pours out, and they weaken the plants. So our job was, okay, where are they going, and what is the danger of the spread of the adults to other places? You know, how far can they fly? We've seen them fly now. We recorded their flights. And I thought if we could see if there was any directionality to their flights, like do they orient to wind and how far are they capable of flying, then maybe we could predict the next place where the population was going to spread to. So that was our thing. But it's pretty clear that when we're working in that vineyard, and there were rules by the Pennsylvania Department of Agriculture that before you left the quarantine zone, you had to inspect your car, inspect your clothes, and look inside and outside the car. But you can see in the few hours we were in this vineyard, on the wheels of my car, you have this. So vehicular spread could be a big deal. So we had to find out about that. During the next year, when there was a mass flight dispersal event of probably millions of these adults near Allentown, my postdoc got stopped by a passing train here in Allentown on the way. He was trying to come to our field site where we were doing video recordings. And there was cars backed up. So he tried to find a way around, but he went into parking lots of a car dealership and saw that there was a Atlantis lining the road, this area, and he watched adult spotted lanternflies. In about 10 minutes, he saw about 50 adult spotted lanternflies flying from the Atlantis and landing on the slow-moving freight cars of this train. That's the only time that I know of anybody has actually observed this. They were hopping on to a passing freight train. And how big of a problem is this? We want to know, are these adult, especially females, are they already mated and then they drop off at the next location and lay eggs, or are they virgin? Important. So this train was heading south, and south of that site is the Norfolk Southern Allentown Terminal. And there's lots of Atlantis here. And these cars could be put on a siding, or they could continue on to parts unknown, maybe down to Virginia, where they've now had an outbreak. So if you look at this initial site of the infestation, and now you look where we are, Center County, but here's Altoona, Pennsylvania, in Blair County. Just this year, I went down there because the Pennsylvania Department of Agriculture found these finds of spotted lanternfly infestation in these spots in downtown Altoona, Pennsylvania. You notice some lines here. Okay, what that is the Norfolk and Southern Railroad Yard in Altoona, way over in central Pennsylvania. And these are the spots where they were found. And you take a look at this down here, this fan-shaped thing. I walked around in here to see whether we might use this as a field site. And I noticed this rail yard right there. And I walked down here, and what this is an engine, railroad engine, repair and maintenance place, and you look at all of uh, uh, different garages for all of these diesel engines. So this is a big rail yard. And my thinking is that, yeah, this lanternfly infestation came down there through the railroad. But apart from the um, vehicular spread, which looks like it can be a big problem, what about flight dispersing females that are flight dispersing naturally? In 2017, we were in some residential areas in one of our field sites. This is site 2, and we happened upon, this is um, a weeping cherry, black cherry, in one residence, and there were hundreds and hundreds of adult spotted lanternflies flying out upwind from the cherry, and some would not make it to the next tree row. They were flying in descending or level flights. Some made it to the tree row. These number ones are all Alanthus, and number 2 is black walnut, and 3 is black locust. So there's a variety of trees in this tree row, but they were seemed to be orienting into the wind when they took off. At another site, site 1, these are white pines. There were hundreds taking off and flying upwind out of these white pines. And I looked, and they weren't feeding on the white pines. They were kind of using them as a way station, it looked like, to take their next flights. And you see, they don't fly that far. We found that they're only flying maybe 50 meters or so upwind, but they have to descend. They don't generate much lift. And we were able to measure their ground speeds. We had pylons here and timed them and measured their ground speeds and air speeds. On the back side of the white pines, the leeward side, you could find no lanternflies taking flight, only on the windward side. And we found that they really don't push more into the wind even at higher wind speeds. So they don't change their air speeds to try to maintain the same ground speed into the wind. They pretty much set their flight motor engine at a certain level and then just take off and fly, because when the wind is higher, their ground speed goes down. So they're not pushing a higher thrust at higher wind speeds to maintain the same ground speed. Otherwise, you would see the line that's level, but their ground speed goes down. So we published a paper on this, 2019. So they take off into the wind, which is a form of anemotaxis, wind steering, but they do this through mechanoreception. They can sense pressure just like we do when we're standing on the ground, and they turn up wind, or a dog with his cold, wet nose can turn up wind using the evaporative cooling on the nose and find the upwind direction. If we use pressure on our body, that's what these lanternflies do to take off. But do they continue to steer with respect to the wind after they take off? So Andy Myrick, on one of the mass flight dispersal days in September of 2017, put his smartphone on the ground and just video recorded from the smartphone the tracks of these lanternflies taking off from Apple and going generally upwind. He is able to digitize these tracks. I won't show the video, but beautiful series of lanternflies flying, 3 meters, 5 meters, 8 meters high. The very tiny ones are flying higher. These guys are flying lower, and able to analyze the wind direction and the steering of the lanternflies using this thing called the triangle of velocities. If you know the track direction and the ground speed, and you know the heading and the airspeed, which you can see because you can see the animal's actual body, so you see which way it's heading, you can calculate the wind direction and the speed and see if they're compensating for the wind by trying to turn more into the wind, which would be upwind anemotaxis. They don't seem to do that. The heading at takeoff is mostly upwind, and the resultant track is a little off the wind line. But so it looks like the adult spotted lanternflies use this, what we call a first aim and then shoot form of anemotaxis. While they're sitting on the plant, they aim upwind due to pressure and then launch themselves into the wind. But they do reiterative bouts of these flight trajectories that are about 50 meters, it seems, per bout. So they progress upwind in a bout by bout process to new feeding sites. And that's our dog Timber barking at the mailman. So we'll just keep going. So how far do they go in each bout when they're flight dispersing in nature? And what else are they orienting to besides the wind at takeoff, if anything? What makes them land and stop dispersing? What makes them land and stop deciding to take another flight bout? Well, during 2018, this is near Allentown. This is Dorney Park, an amusement park. And people were coming down off of the water slides at the top because they walked up there, and there were so many hundreds and thousands of lanternflies up there that they got spooked and they would not ride on the rides. And Dorney Park officials were concerned. And this is some crazy ride. And the lanternflies were up there too. So we went down there, and this was about Labor Day when it was still open, and we scouted it out a little bit. But then after Labor Day, the park closed a few days later. So the lanternflies, of course, were still there. And this was near the Lehigh County Extension Office. There's a Dorney Park, Lehigh County Extension Office. And we worked out of that office. And it's nice parkland that adjoins the amusement park. And we decided, okay, we'll get some females from the ailanthus trees around there, and we'll put them in cages. And then we'll try to release them because they do walk up when you put them on a platform. So what happens when they take flight? Here's a couple of females that have walked up to the top of a ring stand, and they take off. Now the wind was coming generally on those days from this tree row, which has a lot of ailanthus, generally from the northwest or the north, a little bit northeast, northwest, and they would take off and fly off in these directions. And we would see that they could only fly maybe 11 meters for 11 seconds, maybe 21 meters. But sometimes if they could stay level enough, they would get near a lamppost and they would turn to the lamppost and land on it. Or they would land on these garbage cans, okay? And there were little ornamental trees on the side of the parking lot. If they went off that way and they were able to stay level enough, they would make it to the tree and they'd turn and land on the tree trunks, these vertical objects. Or if they went the other direction, if the wind had shifted, they would land on these red maples and usually on the trunks, either any of these trees. So it looked like there was a visual response to vertical objects. And we measured these ones that were taking off from our launch pad, and they could only fly an average of 23 meters in one bout. But when they got on a lamppost or on the tree trunk, they would walk up again and seeming to sample them for possible feeding. And then we assumed they were going to take off again because those weren't host plants. The steel lampposts they couldn't feed on. So we found like almost a third of the adults deviated from their upwind flight paths and oriented to light poles, garbage cans, and tree trunks. The longest flight trajectory we found was 75 meters. So why do they disperse? Well, here's that telephone pole in that vineyard on one day, and we found this astonishing accumulation on the telephone pole. Now, the people I first showed this slide to thought it's an ailanthus tree. Well, it doesn't need to be an ailanthus tree that's a host plant in order to get an accumulation. This is a creosote-laden or originally treated telephone pole from probably 30 years ago, and they accumulate on it at the base, and they walk up the telephone pole um, and launch themselves again. Here are some that are coming in from the vineyard and going to land on the pole. But when you look at these guys on the pole, you can see some of them with their stylets inserted into cracks and crevices in the telephone pole. To me, this is a feeding attempt, sampling, and of course they can't feed on the telephone pole, but they try. So this told us that it seems like these flight dispersal events are related to the need for these adults to find more feeding spots, better feeding spots. And in general, all of these flight dispersal events that we were seeing 2017, 18, 19, and this year are plant-to-plant flights, sometimes very local from one tree flitting down to another, sometimes 50 meters away, sometimes they fly 75 meters, but they're always these plant-to-flight plant-to-plant flights that are to find favorable trees to feed on. To complete their adults' reproductive development is seen that way. And we also found that there's strong visual orientation to vertical objects. They accumulate on inanimate, non-biological things, on light poles, towers, buildings, cars, and this telephone pole that we just saw at Site 4. And when they try to fly off, this visual stimulus from the pole is so strong that they end up landing on the pole again. So it's like it captures them. It's like a trap, like a visual trap. And they accumulate on them and go down to the bottom and then sit there for a while. And then before they try to walk up and fly again. So here's this telephone pole. And you can see in this video, I hope, we took one day, you see all the lanternflies, and a lot of them are coming in, and you see all of them on the pole, and you can see a lot of them walking up. But there's this was a huge flight day, and they're all the way up towards the top. And these guys are on the downwind side. They're flying upwind to the telephone pole. Okay. And if we look at another video of the pole on a different day, and you can see the same thing. The wind is a little different this day, but what you see, see them walking up the pole, and how many hundreds and hundreds of them there are. So the pole is what we called it, the magnetic pole. And we wanted to know how far away from the pole can they see the pole? And whoops, sorry. So Andy took a GoPro camera and he set it down, and this is the original angle of the pole. And he was able, he's a bioengineer, using his skills to take this pole, and then he could digitize the hundreds of flight tracks of these spotted lanternflies. This is the downwind side, so you see them accumulating there from this angle. And you can see a lot of the tracks coming in from the outside and landing on the pole. And he was able to use his skills to take this original angle of the pole and then give us a top-down view of the pole, which to me, I'm just an entomologist, is pretty remarkable. And he was able to then harmonize the various directions of of what was north and what was the wind direction in order to get the tracks digitized. So this is the top view of the pole and where the camera was. We knew the wind direction over the tens of seconds we took this video, and it was only needed tens of seconds. That's north, that's the wind direction. So here's the vineyard before it got decimated. And we were able to see individual flight trajectories. And so here's a lanternfly coming in like this. You can see it like looks like it can see the pole turning, and then it comes upwind and lands on the pole. This one comes by from this direction, from upwind, turns, and then comes upwind to land on the pole. This one comes from a different direction, but comes upwind to land on the pole. So this is the usual way. And you see these heights, 6 meters, 6 meters, 7 meters, 9 meters. The pole is 12.5 meters high. So, and he was able to calculate the altitude from these videos as well. Lots of them flew by the pole without turning. Here's several tracks. They just were flybys. Here's the pole. This one turned away. This one paid no attention, no attention, no attention. So how far away were these guys compared to the ones that turned towards the pole? And you notice these heights, 11 meters, 11 meters, 11 meters, 11 meters, 12 meters. The pole's only 12.5 meters high. So these guys were flying higher, and here's some that were leaving the pole. They climbed up to the top, and you can see they were all flying off downwind, displacing downwind. So we found this, that they will turn towards the pole when they're a mean of 2 meters from the pole, 2.1. And the ones that turn and land on the pole that are 2.1 meters away from it, they're all about halfway below the top of the pole, halfway up the pole. So they had a good view of the top of the pole, what's above them, as well as what was beside them. The ones that flew past the pole were 3.6 meters away from it. So they were farther from it, but they were also only 1 meter from the top of the pole. So the visual stimulus is pretty weak. They have to be pretty close and flying at a lower altitude in order to respond to it. Nevertheless, the pole is a good visual stimulus. It's the only stimulus of any type that I know of that these will give the lanternflies a directed response towards it. No semiochemicals, no auditory signals that we can find, but this visual response to vertical objects is very strong. And we're thinking of using it, for instance, telephone poles along roadways might be an easy thing to sample if you're looking to try to detect the spread of these guys when they're in their, at least when they're in their flight dispersal mode. They're a visual trap. You don't need to stick them on the poles. They stick there themselves for a long time. We need to find out how long, but probably on the order of an hour or several hours, or if it's overnight, you'll find them there in the morning. So could be a good monitoring tool, which we're looking at. We're also working with Brian Walsh in Eastern Pennsylvania for developing maybe attract and kill to vertical objects like telephone poles. So, but what is the threat to the population by these, let's say, the flight dispersing females? Our work in 2017-18 shows that the flying adults are a mixture of males and females, pretty much an even sex ratio, males and females. So are the flying females gravid, and how far can gravid females fly compared to ones that are unmated? And so we published a paper on this recently, again, 2019. So what we knew from 2017 was that the later season females were fat and yellow, and even the later season males were fat and yellow compared to most of the ones earlier in the season, or even during the flight dispersal, are skinnier and black, very little yellow band. Okay, and he's not squeezing them. This is just the way they look. And we're one of the few groups who can actually find mating pairs early on in 2017. So the male's wings are always on top. So here's, we decided to use insect nets to capture the lanternflies as they're flying around from the apple trees to the ailanthus. This is apples to ailanthus, apples to grapes, grapes to apples, apples to oaks across the road. Capture them in flight as they are doing this. And this is a different day. So this is what you see of them flying from apples to this tree row at the edge of the vineyard. Now, it's not like they're flying, this happens to be ailanthus, but they fly this way towards lots of things. Okay, that's a pretty remarkable straight line flight, and the wind is coming from the direction of the ailanthus. Okay, that's just me. That was just my iPhone. But you can see how you can really see the headings, and you could calculate ground speeds and air speeds and all of these things, and the wind direction from these images of these. You can digitize these. This is an amazing ability. These guys are so large that you can measure all these things, and you can't do that with things like aphids and other hemipterans. So we captured these flying adults as they were going from grapes to apples, and we got a bunch of them in this direction. We captured them going this direction. Ailanthus onto grapes, and just like you saw, apples to ailanthus. We stood there, and while they were near us, we swept the net and captured individuals. And then apples to oaks across the road, grapes to oaks, and analyzed their how far they could fly and whether they were fat and yellow. So here's some more mating pairs. You vineyardists will be, this is all on grapes where they got fat, more on grapes. Mating pairs, another mating pair on grapes. So we had captured these ones in nets, the good flyers, going from grapes to ailanthus to oak, apple to grape, et cetera, et cetera. And these ones all weighed less than 300 milligrams, or around 300 milligrams. We captured mating pairs and fat ones from grapes that weren't mating, fat ones from maple, fat ones from ailanthus. And these were poor flyers. We never caught any fat, spontaneously flying lanternflies in nets. We had to look for them on trees, and then we would throw them in the air to see how far they could fly. And they pretty much just fell to the ground. And these were weighed much more than the good flyers. And this is the distances that these fat ones could fly when we jettisoned them, threw them up in the air. And this is, again, how far these distances, the minimum distance, because we netted them before they could fly farther. But this was, they were about to land, the ones going from grape to oak, grape to ailanthus. They were about to land, but these distances were not very great. So plant to plant local dispersal, but it's not bad, 30 meters per flight bout. Okay, but these guys are poor. And they are very heavy, these poor flyers. The black abdomen ones were the ones we got that were good flyers. Are these skinny ones unmated and the fat yellow ones mated? We had to look. So we dissected all of these lanternflies. Luckily, the males pass a proteinaceous sperm packet, like butterflies and moths do. And you can dissect the female and see if there's a sperm packet in there, a spermatophore. And if there is, that means she has mated. And sometimes we found very fresh new spermatophores. We call them fragile. But at any rate, you could declare, yes, this female's mated, or this female's not mated. All the poor flyers and the ones we got from mating pairs, which we didn't try to fly because they were coupled, almost all of these fat yellow females were mated, had a spermatophore. And the thinner, blacker females that were good spontaneous flyers, 95% of them were unmated. That was 5 females out of 100. So in general, if you Ohio people and westward have to deal with this lanternfly, one thing we found is that the mated spermatophore present females, you can tell pretty much if they're mated because they have they're huge yellow area. Okay, that's a general way. The ones that are skinny and black, no spermatophore present. So it's a visual way of deciding, do you have a gravid female or not? I don't know what you're going to do with that, but at any rate, it helps you determine that. So what about these skinny good flyers that are mated, the 5%? What kind of a threat are they? They're as skinny as the unmated good flying females. We're not sure. So this is my summary of the high spontaneously flying, flight dispersing females. Almost all of them are unmated. They will have to land and feed more before locating a mate to become gravid. So they're not very much of a threat. The fat mated yellow females are not going to be able to move from crop to crop. They're going to have to lay their eggs where they are. And they do not participate in these longer distance flight swarms. So they're rooted to wherever they are, where they become mated and fat. These are the ones, these 5% that are mated and skinny enough to fly well, are ones that might be the biggest threat to the spread of the population. However, they are skinny, and they will have to feed more wherever they get to. They have to find a good enough plant that has enough sap to be able to swell up and physiologically be able to have their eggs become mature, is what we think. So they're still not as big a threat as you would think. They would have to find food plant wherever they end up at. So in nature, how far they can go? What makes them land and stop dispersing? So these flight bouts we saw were pretty short because these flying adults can't generate much lift. They can only generate thrust. And so we thought, well, let's see how long they can flap their wings during one flight bout. If they don't have to land because you don't allow them to descend and you allow them to keep beating their wings. So we were able to tether them. This is slow motion at the Lehigh County Extension Office. We did tethering experiments. This is just a thread. Okay, so they will keep flapping their wings if you suspend them like that. We don't need a flight mill, just suspend them in place like this, and you have a fan and you give them enough airflow. They need to have enough anemotactic or wind flow stimulation of their mechanosensors in order to keep flapping their wings. We found that out. But you give them an airflow of over 1.5 meters a second, and they will keep flapping their wings. And some of these single bouts we were able to get in a suspended female, skinny black female, was over 12 minutes. Okay, and some of these females could perform 50 consecutive bouts of flight lasting 20 seconds each. So that's almost 17 minutes of these 50 separate flight bouts. And this explains why, I guess I'll stop my share. It explains why sometimes you get these high level, high altitude aerial, what people call swarms. And these massive high level altitude flight dispersals, we think are just accidental. There are some days when there's more buoyant air, high adiabatic lapse rate, and these ones that are locally disturbing, and you can see there can be a lot of them, they find themselves lifted to high altitudes, 40, 50 meters high, and then we've observed them, and they are carried downwind because there's no visual stimulus up there, and they keep flapping their wings like when we tethered them. And we calculate they can go many kilometers in one of these long flight bouts before maybe the buoyant air isn't so buoyant anymore and they are deposited in a new location. But those days are very rare. Most of the time it's pretty much local, plant to plant, several, maybe 50, 100 meters, and that's it. And then they have to sample whatever they land on, and that takes a long time. And if it is good enough, it's another Atlantis tree, or later in the season, they like red maple. It seems like maybe there's more sap there. They stay there and feed to physiological maturity. Okay, so I'll stop there. Those are some of the things we've learned about the dispersal behavior of adults. And if there's any questions, I'd be happy to answer them.
All right. This has been a very interesting webinar, especially for someone who grows grapes. So thank you for all this information. Whether it's good or bad, we sure appreciate it.
Sorry to show that devastated vineyard, but
if you can't be a great example, you have to be the horrible warning, don't you? So
it's show what can happen. And nursery growers are been pretty hard hit in some areas. Nursery trees.
Okay, we have a question here. What is your definition of established populations? Breeding and egg laying, or multiple stages found together, nymphs and adults?
When you find egg laying, I think is the standard.
Okay.
I mean, or if you find, any early nymphs, that means that you're not just getting a fly-in adult. Or at Penn State, one of the years in the parking lot near the football stadium, a dead spotted lanternfly was found. So it turns out it just had fallen out of somebody's car. It had been dead. So, but it was a find with no established population. So just because you find an adult doesn't mean it's established.
All right. Could you share your opinion about the relative dispersal threat posed by egg mass flights of gravid females and the 5% skinny females?
Yeah, so egg masses, definitely, it's like gypsy moth. I think that's what people worry about, and especially like on trains. So, or cars, construction, or RVs, just like gypsy moths. One of these gravid females could conceivably crawl, even though they can't really fly. They could crawl to that object, and they will crawl towards vertical objects and deposit their eggs on the steel wheels of a train car or a freight car. And they will lay their eggs on anything, which is another reason that, they're not very host specific, even though you find them on Atlantis a lot. They lay eggs on anything, rusty, discarded objects. So an egg mass could be deposited on a car or a train and carried to parts unknown and sit there over the winter on a siding and then the first instars come out and they will hop. They're capable of hopping tens of meters, and they will feed on any little grasses and things. So they don't need trees. They feed on little succulents, and they could develop further and hop to bigger trees and become adults. So egg mass is definitely a big worry, especially on things that sit for a long time, travel long distances, and then sit somewhere a long time into the next year. The gravid adults, yes, the big fat adults could walk and climb onto a car, a train car, for instance, and get carried. We don't know that the rail yards are pretty big, and they would have to walk a long distance to get there. The 5%, the skinny females that are mated, um, it depends which is the bigger threat, vehicular transport, which I think is probably the bigger threat in general, or the natural flight dispersal. If one of those skinny black mated females was able to fly onto a train car, yeah, then it could travel, for instance, south to Virginia and find an ailanthus tree to complete its development. And if there was males on those trees, they could find a mate after they plumped up with feeding. So that's all I would guess.
Okay. What methods did you use to try and measure sexual pheromones and audio cues?
We used a vibrometer, a little phonograph needle, with a recording device. This is a standard way. People have done this for years. Now they use laser interferometers. The thing is, the signal, first of all, you don't see any behavioral directed movement of a male to the female or vice versa when you watch these guys for hundreds of hours. So despite the lack of seeing such directed movement, which would mean if they are emitting a sound, it's not a signal because you're not getting a directed movement. We tried to put the vibrometer on the tree trunks. We're in a quarantine area, and out of the quarantine zone, so we couldn't have this in the lab, in a soundproof lab, et cetera, et cetera. But first of all, you have to find virgin males and females. Rearing is a problem. But at any rate, we put them on trees, and there's just background noise, and you get waveforms and signals, but you don't know the communication value, and you don't see any behavioral evidence of any directed movement. So that's what we use for that. We tried extracting adults in 2015 and 16, and again, you need a bioassay. You need a behavioral measuring stick for, is what you've extracted from the females, does it have signal value? Well, if you don't get any directed movement reliably, even when you're watching the wild males and females, you have extract and you have compounds, but there's no way to see if any of those are, quote, attractive, because nothing naturally, you don't see any behavioral evidence of attraction in the field, males to females or vice versa. And there's people who say that there are like a 3-component blend of volatiles from ailanthus, but that's only been shown to work in a laboratory. The field data is arguable. People I know, and these compounds aren't ailanthus specific. They are just general plant volatiles, terpenes, some green volatiles. And besides, you don't see, if you watch these guys, you don't see directed movement towards ailanthus over any other tree. You can see them flying from ailanthus to black walnut, ailanthus to some foliage, herbaceous layer, flying from Atlantis to ash. We do have some ash remaining. They go any direction. They fly off of Atlantis as often as they fly onto it. They do seem to accumulate on Atlantis during a large part of the season. I use accumulate, not aggregate, because I think it's just that the sap level, the turgor pressure on some of these trees is much greater than on other trees. Even neighboring Atlantis are avoided. Um, and they accumulate on one Atlantis out of a bunch of them, and they stay there. And I think that the turgor pressure, these guys need a lot of force from the sap to get it into their stylets and through their digestive system. So that's why like I said, the only thing we see that's a stimulus that causes a directed response towards it are vertical objects, and it seems to be visual.
Okay, so in other words, Atlantis, they could develop without Atlantis being in the area, correct? Or
That is, yes, that's the current thinking. I mean, they're some people have said they start on, well, they can develop right through on grapes. I mean, they can develop on Atlantis. They spend a lot of time on black walnut, for instance. But then later in the season, right now, after these years of research on it, not by me, but by others, maples seem at the end of the season to be the preferred place to finish development. And probably has to do with the level of the phenology of the trees, and the red maple might have more turgor pressure, more nutrition at the end of the season.
Okay, you may have answered this, but I'm going to read it anyway. Since this was asked a little earlier in the presentation, since they don't produce much lift, if they don't make it to the next vertical object and end up on the ground, do they then walk to the nearest thing to climb?
Yes.
Okay.
Yeah, I mean, they fly. This is a problem with even trying to observe them. You're in the middle of a lawn and watching them, and they will turn and land on you. And this happens to everybody. I mean, they land on people, vertical objects. And if they land, I've seen it, and so have my colleagues, once they land next to you, a few feet from you, and you stand stationary, through the lawn, they will walk and hop and come to your feet and your legs, and it will climb up you just like you're a telephone pole. So they walk towards vertical objects as well as fly. Mm-hmm.
Okay. If attempting to use telephone poles as attractant traps, I'm assuming the phone company and their repair workers would strongly prefer you use vacuum traps to painting the poles with sticky.
Yeah, no, we're not anticipating. First of all, there's two things. The telephone poles, and this is all pie in the sky right now, but the telephone poles, you don't need to put anything on. You just need a scout, let's say, in this scenario, and you can drive along a road very fast and like stop every mile and examine a couple of poles, because the lanternflies stay there for a long time. No stick 'em, no anything. And they walk up to the top and they stay there a long time, or various. So we don't know the scheme for trying to visually sample the pole, but it's just they're already there. There's no expense in putting in traps or anything, because these visual traps are already deployed all over the country. Okay, that's the idea. And they are an arrestant. They stop the animal for several hours probably. The other pole type of thing would be an object you installed like around a vineyard. And there's various ways to put toxins on such things. There's already toxic netting for the bed netting used in Africa, and this is what Brian Walsh is experimenting with down in near Reading, and having good success in killing thousands and thousands of adult spotted lanternflies in nurseries. So the idea would be to deploy attract and kill objects like that, and you don't need to spray the poles. You put toxic netting over them, and so far this is showing some real good promise. Of course, there's a problem. How many poles do you need? What's the scheme? How far apart? Blah blah blah. Early days yet for this.
Okay. Are males preferentially drawn to skinny black females as unmated, and sterilized ones might be a good trap population, like medfly control?
Yeah, that's good. And then first, they don't fly far. So anyway, the male, nobody. We're the only group that's seen an actual courtship, and that's one out of several years of watching. It takes 3 seconds, and the male happens to be next to the female, and these are fat. They're already fat. I don't know how the skinny ones get mated, those 5%. I don't know. But we've only seen one with a swollen male, swollen female, yellow, and it took 3 seconds, and it was just like a quick wing vibration, bang, and they're mating. Now you can more easily find copulating pairs, because we've seen that they stay copulated from 2 to 4 hours. You can find them in couple for hours, but the actual behavioral courtship sequence is so fast, and you can't rear them to get enough virgins to see skinny ones versus fat ones. And yeah, that would be good if you could do that, but lots of questions. And then sterilizing, doing sterile male, but these sterile insect projects that have been successful, they don't just sterilize males, they put everybody through the cobalt, radioactive cobalt in whatever they're using. And then they disperse millions of them via airplane, and dump them all over the place. And we can't rear that many. People have had a hard time rearing enough to do some simple experiments.
Okay. Rachel says, a great talk. Is there any evidence that non-orchard trees are negatively affected by spotted lanternfly feeding?
Non-orchard trees, I mean, that I don't know. That's not my bag. But non-orchard tree is Atlantis, and they love Atlantis. Things that they don't like. I don't think there's any evidence of like a semiochemical deterrent, a feeding substance, secondary plant substance that they don't like. I think the weight of the evidence is that anything that gives them enough sap at a strong enough pressure, they will stay and feed on that for a while at least.
Okay, if you find spotted lanternfly on your property, should it be sprayed with pesticide?
Injection with what's the dinitrophuron, it's a systemic. That's how the PDA and USDA that's how they initially started. They used trap trees to some Atlantis that they were in an area, and they treated them with this dinitrophuron and killed tons of lanternflies, and then cut them down. So the homeowner, yes, could protect a tree, with a systemic insecticide like that. And I know Brian Walsh, he's a nursery IPM guy, had his own private business. He protected many homeowners' trees with this systemic.
Okay. You mentioned a minimum wind speed, 1.5 meters per
Mhm.
second, as one factor needed to cause adults to fly. Is there any other factor, such as a density on a tree or pole, that is also needed to initiate their flights? You
Yeah.
mentioned. Oh, I'm sorry.
Mhm.
Go
Go
ahead. You
ahead.
can answer that, or then I've got
No,
another one
keep
here.
going. Let me hear the whole thing.
Okay. You mentioned locusts casually, which have a density-driven life
Mhm.
cycle / development. Is there something like this for spotted lanternfly to stimulate flight?
Mhm. Good question. Of course, the locusts, they have a, they become, what's the word? Anyway, there's a solitary phase and gregarious phase, and there seems to be a pheromone involved in getting the gregarious phase together. But that's just large numbers, and they get physiologically changed by the large numbers. And they become more flight active. In the lanternfly, we think that there's not such gregarization, if you will, that the densities, the accumulations on the trees in the area, and that's why we didn't see these mass flight dispersals in ground zero and the initial infestation area until a couple of years had elapsed. And we think they've been feeding on the same ailanthus trees. And you could, in 2017, you could see a lot of these ailanthus trees were all browned and had a lot of flagging, which we didn't see in the earlier years. So we feel that the food resource by these thousands and thousands of lanternflies feeding on the same trees had become insufficient to allow them to develop physiologically. So this physiological stimulus would be insufficient nutrition. And then they start becoming more active and crawling up from the trunks of these trees where they were used to feeding in previous years and get to a high point, and they always crawl up till they get to a very top of whatever they had, and they launch. So we think it's this poor nutrition more than anything. And you sometimes see them bump into each other, like on the telephone pole, and it's like a version. It's an accidental, um, not defense, but they just take off for a bit, but they land back on the same thing. So there's that kind of stimulation. But it's generally, they get more active when they haven't had enough to eat. That's what we think. And then they go to new zones where there's, or they even locally fly 100 meters, land on something, and hey, maybe that tree has enough of sap and turgor pressure.
Okay, how effective do you think egg mass survey would be to help determine population density? Would egg mass destruction be useful or even possible in reducing the spread of a population?
Yeah, I'm not the certainly an expert on that, and I think that's encouraged. And in the early years of the infestation, when it was small, people were encouraged to scrape egg masses and find other ways of, there's all sorts of anecdotal things about using, laundry detergent sprays, whatever, what works to kill egg masses, scraping. And there was a lot of effort by even the PDA to scrape egg masses. The thing is, they lay eggs on anything. One of Erica Smyers, who I showed in that study, when she did an egg mass degree day study on the property she was monitoring natural egg masses, she had many that were under rocks, under boulders, um, that hatched after so many degree days, as well as ones that were on Atlantis, the ones you end up scouting for. You know, okay, let's look for egg masses on Atlantis, but they're like everywhere. So I would say in the earliest days of an infestation, when there aren't many egg masses, probably yes, killing egg masses could be effective in slowing the spread or the development. But once you have high populations, it's you know, I think it's you got to do other things.
Okay. We have one here. Is there any hope for control of these insects? If so, what? I think you kind of covered that. And I think isn't there a website? Penn State doesn't it have a website that also talks somewhat about control?
Yeah, I think so, and that's why I can't say that I've looked at it recently, but I got my head down working on my area where I think I can contribute that other people aren't working on. So I'm filling a niche in my area, and I've got to keep my head down and try to answer these questions about this kind of dispersal. But yes, this massive USDA FIFRA SCRI grant that Julie Urban is heading up has a lot of players at different universities: Cornell, Delaware, Maryland, Penn State. I'm not sure, maybe Michigan State. I can't remember, but the people are looking at pathogens. They're looking at biocontrol. Um, it would be something like that, probably some spotted lanternfly pandemic that you could initiate. That probably could hold the best chance right now. Again, the spread of a pandemic of a pathogen, you need a lot of this. They need to disperse and spread the disease. So who knows how long that would take. So it's biocontrol. People have been searching, and there are some natural enemies that have popped up and are in quarantine in the USDA. But I think I think it would just have to die out from some kind of pathogen.
Okay, and I will look. I know you're right about yeah. Michigan State I think is also involved in that as well. So I'm going to look up that website. To all of you who are asking these questions about controlling the insects and that kind of thing, I will look that website up and the links and put that on the email that you will be getting tomorrow. So that might be helpful for all of you instead of trying to ask all these questions about that and I
Yeah,
think that'll be a way to go.
there are experts in these different areas that I can't answer. So
Okay. Oh, and boy, my colleagues in EAB University are already ahead of me here. I scrolled down and see both Amy Stone and Cliff Sadoff have put that website on the chat pod. So you have it there, and I will also put it on the email that you all are getting. Hold on just a second here. Oh, here we go. Is there any signs of bird feeding on the spotted lanternfly?
yeah, it's few and far between. Although I know my colleague Kelly Hoover has started a citizen science thing about having people watch to see if they can see how frequently birds feed on these things. That's good. I think should be looked at, but people have been loath to, or been very. It's been hard to find anything that feeds reliably on these bugs. Birds, stink bug predator. You see occasionally we've seen a stink bug feeding on one of the nymphs or adults. It's very, very rare. Birds I've never seen. You see hymenopterans flying around the honeydew all the time, but they're feeding on the honeydew and not catching the spotted lanternflies and feeding it to their offspring, anything like that. Now these bugs are protected by the chemical defense. That's why they're aposematically colored. They're so beautiful, pretty. And they have at least one thing in there is a lanthone, which has been extracted from Alanthus. And I'll tell you an anecdotal story. The one residence I was working at in 2017, I had a nice little dog, and the adult lanternflies were in a flight dispersal mode, and that's why I was there watching them and collecting them. They were all over the lawn because they can't make it to the next high object. They were landing on the lawn. And the woman there said 2 days ago, her little dog, nice little dog, she let this dog out all the time to run freely in the yard. Well, the dog came back in and one day and laid down on the living room carpet and threw up. And in the throw up was 12 adult spotted lanternflies.
No.
For some reason, this dog ate the 12 spotted lanternflies. And that dog pretty much laid there for 2 days, she said, and was really sick for 2 days. And then it finally recovered. It's anecdotal, but so a vertebrate predator, I would imagine a bird would be affected in the same way. I mean, you might see a bird feeding on the adult lanternfly, but if they fly away, you don't know if they died or if they threw up or whatever. So you need observations like that to see how palatable these things are to a bird or anything else, or a stink bug or anything. Just because you see feeding doesn't mean it's a good thing.
Okay, we have one more question here. This is the last one for you, and anyone else who's still on the call, the webinar, please, I will be sending you a link to, and when I send you the email, to Dr. Baker's email address. So if you have questions, you can send them his way on his email. This question is there a, excuse me, is there a difference in the spermatophores of skinny females? Are they less fertile than the fat ones?
Yeah. In the skinny females, I have to say, I think 4 of the 5 were these fragile ones that were like not completely developed and hardened, tanned. I don't know what the significance of that is. I mean, we killed them, we dissected them. So fertility, it's a good question. Don't know. We don't know why spermatophores are fragile, and we just think they're fresh, but maybe the mating was cut off by the skinny female before the sperm itself was put into the spermatophore, or what. So that, yeah, could be a line of research. Again, you need to have a colony of these things to study that way. So tough.
Yeah. Well, I'm getting thank you from participants saying thanks for all that you're doing with this, and they're saying this has been an awesome webinar, and thank you for all your help with this. So, and I would also echo that. We're so glad that you were able to share your information with us on Emerald Ash Borer University. And again, we've got this recorded, folks, and it will be available on the emeraldashborer.info website on the EAB University page, either later today or first, depending on kind of how YouTube will finally get it put together, or tomorrow sometime. So with that, thanks again, Dr. Baker, and everyone. Thanks for your participation, and everyone have a good day. I'll end the webinar now.
Okay, well, thanks for having me, Robin. And
our pleasure. Again. Yes, thank you.
Bye-bye.